How's everybody's weekend?
Good.
It's good. What is the best thing that you ate this weekend?
Chicken Alfredo.
Ooh, made it or bought it?
I grilled the chicken. And
You grow the chicken?
No, I know. I grilled the chicken.
Grilled.
Yeah.
I thought you said grew the chicken.
No.
I was like, that's dedication there.
And then my mom made the noodles and the sauce. And
Lovely. Awesome. Thank you. What else? What's the best thing you ate this weekend? Girl.
Chocolate chip cookies.
Ooh, where'd you get them? Did you make them? Make them bar.
Mason campus, like the dining hall, they have chocolate chip
Oh,
cookies.
cool.
They're really good.
Nice.
Yeah.
Nice. Perfect. Cool. Awesome. When was that? Saturday? Friday?
Saturday.
Cool. Go ahead.
Ice cream.
Ice cream. Make it or bought it? Bought? Where at?
No,
Grocery store? Or
7-Eleven.
7-Eleven? Ooh, yum. Ooh, yum. Very good. Anybody else? What else? Did you have?
Dumplings.
Dumplings. Make them or buy them?
Make it.
Did you make it? Nice.
My wife.
Lovely. Would she?
It's called the jiaozi.
Jiaozi.
Yeah, make it,
Oh.
beautiful.
Would she be interested in making some for the
Yes.
for all of us?
I have a PPT presentation on how to make that.
Oh, cool.
Yeah.
We might listen to that. We might listen to that if you're willing to offer it one of these days. Perfect.
Yeah.
I build into the schedule because we don't do exams, right? There's a couple of days where it's like I sort of build in buffers. We might do. I've done for the midterm last year. We did show and tell. It was like bring in anything and you have 5 minutes. Somebody like back in the pandemic when it was in full raging, and everything was on Zoom for show and tell. Somebody brought in their 4-year-old niece. There were dogs, cats, pets, family members. I'd like to show and tell. This is my niece. Cute. It's hard to top that. It was fun. But yeah, that'd be great. That sounds fantastic. That sounds fantastic. What else? Best thing you ate this weekend? Sylvie.
I made risotto with shrimp,
Ooh.
and I made the shrimp. I made the broth using the shrimp shells and
You like some
veggies.
shrimp stock? Yum.
And I also put in the risotto like a shrimp bisque that I made.
You say risotto better than I do.
Risotto.
Yeah, because I'm from Missouri, so it has a D in it. Risotto.
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 I had an argument once with a friend of mine whether biscotti is because he pronounces it biscotte. I was like, I think it's biscotti. So it's like he drops the I off the end of a lot of things. It's interesting. So it's like, I don't know. We'll talk one of these days. Awesome.
Do you say bruschetta or bruschetta?
I've heard it both ways, and I've heard, my friend calls it bruschetta, and I said bruschetta. It's like, I say it like they say it at the Olive Garden,
Don't go to Olive Garden.
I try not to, whenever possible. But, yeah, food's weird.
What's the thing? What's
Pardon me?
that? What's that? The thing?
Sylvie?
No. The thing that you guys were arguing about.
Was that an
The
argument?
words.
Oh, yeah.
No. With your
Pronunciation.
Sorry, just
Like what was it?
No,
No, what was the argument?
What is bruschetta?
Oh, it's like this little like a slice of baguette with tomato, onions, and I think peanut butter.
Like tomato basil.
It's really
That is really good. And there's yeah.
Anything on top.
Yeah, and there's discussion on what to do with the bread. Is like you slice it because I think you're supposed to like flame grill it, like over a live flame on either side to get a kind of a burny, charcoaly flavor to it. But you can put it in the toaster before, so one does what one can with what one has, right? One does what one can with what one has, right, Reem? What's the best thing you ate this weekend?
Oh, I made myself some summer stir fry.
Ooh, tasty.
Instead of shrimp, I use grilled chicken.
Yum.
Yeah.
Yum.
This is the only way I'll eat cherry tomatoes. Regular tomatoes.
Yeah.
I can't.
What'd you have, Reem?
I ate lunch ice cream.
What'd you have? Matcha ice cream? Yeah, that's good. Where'd you get it? Awesome. Name of the shop. Okay. Yeah, matcha ice cream is super good. Super good. I don't get it often when I do. It's like, man, that's good. What?
You tried to get it.
Yeah. I'll say whatever every Westerner says who's not into K-pop. The texture's weird.
You think so?
A little.
Oh, I think it's-
Does anybody think the texture of
Jelly.
mochi is
ice
weird?
cream is like-
Like it tastes good. Everything's like, the texture's weird.
like
I
it.
still enjoy it, but I don't get it often. It's like, the texture's weird.
Have you ever tried chocolate eclairs?
Yeah, oh yeah. Yeah. Do you see
You guys are
this?
making
Do
me
you
so
see this?
hungry.
Yeah. Yeah, I have. Not often, but recently. Yesterday, I enjoyed for the first time this little Azerbaijani shop down here on Pickett, Baku. Had some tea in there, really good. Azerbaijani pastries. This is a place where I can eat too much. That's what I thought to myself. Very good. Well, then, you're fueled for the day. I'm not getting out anything that's going to be traumatizing this one. Okay. Twice so far, I've given you a document that looks kind of like this. And it's like, I keep intending to get to it. Oh, welcome. Hey, what's going on, girl? What's the best thing you ate this weekend?
What?
What's the best thing you ate this weekend?
I don't even know what I ate this weekend. Like Sarku. Yeah.
Yum. It looks kind of like this, and I don't know if you bring things back that I give you in class. The one time that I've done that, I swear we will get to this today, and it will blow your mind. This week's lab is about enzymes and how enzymes work, and I brought a bunch just in case we needed some, because I realized that I first gave this out to you about 2 weeks ago, and it's like, I don't know if you have them or not, no harm, no foul. Enzymes and how they work. And we'll get more about this on Thursday as lab sort of gets closer, but for now, we want to do some of the back-end mechanics. And we're going to do the back-end mechanics, Ms. Muho, about how enzymes work by going through nucleotides, our fourth macromolecule. Sound good?
Okay.
You need one? Yeah, come on. Yeah, come up and grab one if you need. You'll very briefly be on YouTube, but that's okay. I have 2 other pieces of personal news, all related to my strange habit of this semester. My foot-long eraser has come in, but for 2 days in a row, I've forgotten it. It's still in my condo. And we'll get there. And the other piece of news is that I picked up a brand new chalk that I've heard legends about that is supposed to be pretty good. It is the Dixon Railroad chalk. I mean, it weighs nothing. It comes in a box of 60, so I have this case of chalk in my office, about that big, because each piece is like this. But it writes.
Nice. Whoa. It sounds like your other job.
Great. It writes great. What I'm realizing is that when I'm calling good chalk, a lot of places refer to as chalk. What I call lousy chalk is Crayola chalk. So, it's like most chalk is actually pretty good. Most chalk is actually pretty
What's
good.
chalk? Like, what is chalk?
And I did get the Horogomo Full Touch color series if we ever need that too.
Can I try it? Thank you for answering.
Can you try it? What, the fancy chalk?
Yeah, you've been talking about it a lot.
We'll see if we can come up with a reason to get you up here in the front of the room to do something fun. It doesn't erase as cleanly as the Horogomo chalk. What are you gonna do? Okay. I'm gonna get a new piece. This one's down to the
That's not very eco-friendly.
nub, and I will take a piece randomly out of the box. Okay. What did I say we were gonna do? Acids via nucleic acids. There are, usually when we think about nucleic acids and nucleotides, we think, oh, I mean, the obvious thing, you say, hey, DNA, perfect. What do you do with it? What do you do with it? What is it good for? What did they say in high school? DNA is the proteins are the building blocks. DNA is the blueprint of life. Mitochondria are the powerhouse, right? Gregor Mendel is the father of genetics. Natural selection is survival of the physics. None of these things are true. Store information. With it, you store information. For occasional use. More about that later. Properties of it. It's double-stranded. Double-stranded. There is complementary base pairing between the two strands. There are 4 nucleotides found in DNA: adenine, thymine, guanine, cytosine. When we get this complementary base pairing, adenine base pairs with thymine, guanine base pairs with cytosine. And vice versa. So when you see an A, you put a T. Where you see a T, you put an A. Where you see a C, you put a G. Where you see a G, you put a C as the base.
Is it bad if like one of the pairs, like one of each pair's partners up with one of the opposite pairs? Like if A partnered up with B?
Call that a mutation, right? A mispair. A lot of times when we mispair, an enzyme will swoop in on the back end and repair it. A lot of times it doesn't, or occasionally it doesn't. That can either be meaningless, trivial, where there are not consequences depending on where the mutation happens, or it can be miscoding of a protein, or it can be catastrophic and the embryo miscarries at 16 days or something like that, 16 cells, if it's like super bad. It all depends. Speak.
What's the next word? What's next to strand? Like a B.
This word?
Next to.
There's like there isn't one. Between, BTWN. It's shorthand for between, BTWN.
Thank you.
But you learned all this in high school, right? Yes. Cool. Awesome. What our trick is going to be is to understand by the end of the day how these rules of complementary base pairing and double-strandedness provide the basis for information storage, and how the strings of complementary base paired nucleotides ultimately results in something that has biological usefulness. So how do you get from a strand of DNA to something useful? That's our question. With me? Perfect. So if you have a strand, the complementary strand would have which nucleotides on it?
T, A, G,
Sorry, G, correct?
C, G.
Skills check number 1. So when we drew nucleotides, do you remember what these nucleotides look like? They're like a ribose sugar with like this other thing coming off of it with like another to sometimes more like OHs coming off of it in all kinds of wonky ways. All kinds of wild stuff. A phosphate group that this is like the worst nucleotide ever drawn. Like, don't try to even write this down because it's like there are like 9 things wrong with it. But they're not trivial. I mean, they're these kind of bigger, complicated things. The molecule itself, DNA, deoxy. So it's the deoxy part refers to one of the side groups as a hydrogen, not a hydroxy group. So it's deoxygenated. A ribonucleic acid. The ribo referring to ribose. This part right here. Look at it, it's a ribose sugar, right? So the basis of a nucleotide is ultimately a carbohydrate, right? Yes? The basis of a triglyceride is, remember the glycerin molecule at the top? Is a remember glycerol molecule? Remember glycerol for a triglyceride?
Oh,
What is this?
carbon.
Yeah, it's a carbohydrate. It's a carbohydrate. So the basis of a triglyceride, the glycerol is the basis of a DNA or a nucleotide. It's a carbohydrate. So it's like the basis of a carbohydrate is, give the easy answer. A carbohydrate. So it's like the basis of 3 of the 4 macromolecules are carbohydrates. Go ahead.
Totally unrelated, but does the carbon part of carbohydrate stand for the carbon?
Of course not. No.
Oh,
They're made of the raw material carbon dioxide, but it's like I've never heard that. I don't know. There might be some of the Latin root or something. It beats me. We can look it up. When I say of course not, what I mean by that is possibly. But it seems odd to me that would, because they're all carbon E, so it doesn't provide distinctiveness. But maybe. Yeah, I wonder too. So triglyceride, glycerol, carbohydrate. Nucleotide, basis of it is a ribose sugar. It's carbohydrate with weird stuff added. Triglyceride, it's carbohydrate with weird stuff added. Carbohydrate, it's carbohydrate with no stuff added. Cool? Put more on the board today than many people do in a semester. I'll immortalize this in photograph form. One more thing. So, if it's like, here's one, here's one, here's one, here's one, right? As we go down the line. These bonds here that combine the nucleotides down the DNA chain are covalent bonds. The bonds that attach the base pair nucleotides to each other are hydrogen bonds. So the nucleotides are bonded very strongly to each other down the chain. They're bonded very weakly to each other across the chain. So it's like the strongest bond goes down the chain, the weakest bond goes across the chain, which has implications for how it is used. If we want to access the information, we can't do it from the outside of the chain. We have to do it from the inside of the chain, which means eventually, when it comes down to putting these things to actual work and we have to use it, we have to split that down the middle, which we can do if you have a weak bond here. So you don't want to covalently bond these nucleotides to each other, because then you wouldn't be able to separate the strands to get the information out. It's like if you opened if you have a magazine and you're flipping through it, like we used to do back in the 60s, the pages are covalently bonded to each other, like on the page, but they're hydrogen bonded to each other from page to page. So it's like when you turn a page, the page doesn't rip, because the structure inside of the page is greater than the structure between the page. DNA is the same way, right? So they're loosely attached to each other, but they're strongly attached down the chain. So it's like a zipper. You could easily just grab the top of this and you could pull this apart to get into the information on the inside. Question, David?
What the word? Is this a covalent?
Yeah, these are the covalent bonds here. Is attaching one nucleotide to the other. It's covalent this way. It is hydrogen this way. And that's super important. Bless you, my child. Does that make sense? Perfect. This bond here, because we always talk about what kind of bond it is that we use to attach monomers to each other. These are referred to as the phosphate functional group. This is a phosphodiester bond. Phosphodiester bond. So what do we use to attach amino acids to each other? We use peptide bonds. Here we use phosphodiester bonds. With carbohydrates, we use glycosidic linkages, right? In college, you learn big words sometimes. It convinces the people that are paying for your education that you're learning something. It's worth doing. The MCAT's going to have big words on it, so pay attention. Good news is too, this stuff does come up. Where are my erasers? Remember this nonsense we were doing over here last week? And it's like, I can't even look at this thing any longer. We were making this thing to look like a chart of different kind of bond. Yeah, we can finish that up now. It doesn't erase as well. The railroad chart. Carbs combine to each other with glycosidic linkage. Amino acids with peptide bonds. These glycosidic linkages look like that. Peptide bonds look like that. Phosphodiester bonds with the nucleotides. Like that. A terminal carbon to an oxygen to the phosphorus functional group to the other carbon. Phosphodiester bonds. And triglycerides, how do they bond together? Well, they really don't make polymers. So it's just like they make bonds. What can I say? Go ahead.
So what's the difference between like the covalent bonds and phospho?
These are covalent bonds, but this type of bond is a phosphodiester bond. It's where you use a phosphate functional group to combine two monomers into a polymer. This is a covalent bond between carbon and nitrogen. This is a covalent bond where you link a carbon to an oxygen to the next carbon down the line. Yeah.
Is that a carbon on the bonds?
Which one?
On for phosphodiester.
It's a carbon of the one nucleotide bonded to an oxygen, bonded to a phosphate functional group, bonded to another carbon.
Okay, I just didn't know that was like a parentheses.
So it's covalent bonds all around, right? But you get these typologies. So it's like, here's the phosphodiester, here's the peptide, here's the glycosidic linkage. So not only do these macromolecules have like these curious internal curiosities, like 3 of the 4 of them are based around a carbohydrate in one way or another, which makes sense considering the prevalence of carbohydrates out there. What does photosynthesis as a reaction produce?
What's the word?
What is photosynthesis? What is the output of photosynthesis?
Sugar.
Yeah, carbohydrates, oxygen. So it's like, there's tons of it out there. Of course, it's going to be the basis of the greater structure as it emerges on the top. Of course it is, right? That's one of the first things we get in nature, in evolutionary history, is the carbohydrate. So of course it's the basis for all this stuff. Makes sense to me. Makes sense to me. I am living proof that you don't have to be an artist to be a biology teacher. Go ahead.
What is that word there at the end?
Bonds,
I don't know the other one.
triglyc.
Oh.
I got tired of writing it, so I just stopped.
The last letter is C.
Yeah, there should be an uride underneath it, but I got tired of writing, so I stopped. It's like, you know why? You know why I stopped writing? We've been there. We know.
So like the triglycerides make the bonds?
The triglycerides are held together with bonds.
Oh.
There's no weird typology with the triglyceride. It's just covalent bonds. You just get it by combining a hydroxy group to our carboxyl of a fatty acid, and you get a bond in water, right? Like you always do with dehydration. Questions, thoughts, comments? Cool. I mean, this one's sort of nonsense. We don't even need to have this last one up there because it's not like a different, unique kind of sort of bond. It's just like these 3, however, are distinctive. So we have our DNA molecule. What we're going to do functionally from that is make a molecule of RNA. RNA. Ribonucleic acid. Single-stranded. Complementary base paired. Ish. Get more of this in just a second. We have to make our table of how complementary base pairings a little bit differently than we did before. Single strand, complementary base pair. Shortest strands. Because it's a little more unstable. It's a little more unstable. I mean, you don't get these huge, gigantic, 3,000, 4,000 base long RNA strands because they get weird and breaky and fragile, so. Single-stranded, a lot shorter. And it is,
Did I say unstable?
yeah, they're sort of unstable. I don't want to put too much in the word unstable. I'm going to say they're kind of breaky. They're a little fragile. They're a little fragile. So, I mean, they're a little fragile because they are not double-stranded. That double-strandedness provides stability because it's just more infrastructure, more structure in place to hold the thing together. Single-stranded don't have that, so it's just kind of breaky. So you get short strands. And it's coded from the DNA molecule. So if we have a molecule of DNA, and we have our 4 nucleotides, the other strand of DNA would be
A, Z.
C. Perfect. If we then say, you know what, I'm going to actually use this DNA and make a molecule of RNA from it, going to split it, and we'll take that strand for now. We could make a RNA strand from either one. Okay, but I'm going to use this one for now, just for ease of use. These are A, C, T, G. What color is RNA? Blue? Orange? Orange.
Orange. It's spooky season.
It is spooky season.
You.
Slow down there, Tex. The RNA strand would be U. You get a different one. It's called uracil. Uracil. C would be G is the same, A is the same, C is the same. The difference is when you get an adenine in DNA, that's got a complementary pair to a uracil in RNA.
Okay.
Easy enough. Do you remember that? So if you see just like a strand, like just existing floating in space, like a single strand, you can always tell whether it's a strand of DNA or a strand of RNA. If it has thymine in it, it's a strand of DNA. Thymine is not present in RNA. Uracil is the equivalent. Perfect. Makes sense? Awesome. Which brings us to this. Anybody have one of these or something similar? First base is, I'm going to break that into a 3. I'm going to call this triplet right here a codon. First base is U. Second base is G. Third base is A. What amino acid does that represent?
Stop.
U, G, A. Stop. It's a stop codon. Every single protein you make in your body, like all of them, like of the 25,000, have at its basis an RNA strand that accompanies it that is stored longer term information wise in your DNA. For every one of these protons, the last little DNA fragment, the last 3 nucleotides of it are always going to be a stop codon. Is there a start codon? Is there a start? What's the start?
AUG.
So AUG. So every messenger RNA that you have that starts with AUG is going to be a start. So in the DNA, where you see a T, A, C, everywhere you see your DNA where it's a TAC, that's going to be the start of a protein. Anytime you see an UGA or one of the other twos, that's the end of the protein. The stuff in the middle is the information. So, so here's a DNA chain. What is the RNA chain going to be? And here's the other strand is up at the top. Let's just say we're just going to read off of this one, right? So our RNA strand is going to be
A, U, G, 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 C, U, U, G, A, G, U, G, A.
amino acid. What's AUG?
Start.
GCG.
Glycine.
Sorry, GGC.
Glycine.
Glycine. UUG.
Leucine.
Leucine. AAG.
Leucine.
L.
Lysine. Lysine.
Oh, okay. Lysine. UGA.
Stop.
You just learned how to genetically engineer things. This is now primary protein sequence. It's then going to contort into secondary coils and folds. It's then going to conform into a 3D protein that is either structural or working as an enzyme. Go ahead.
What does the AA stand for?
Amino acid.
Oh, okay.
Amino acid, yeah. So our nucleotide conversation turned into a protein conversation. Yes.
Yes.
Remember this secondary, tertiary structure? Remember that from last time? Very cool. So what this means is that if you know a DNA or an RNA sequence from a COVID-19 spike protein, you can go ahead and take that messenger RNA, put it in a little fat bubble, inject it into my left arm muscle, and it will go enter a cell somewhere throughout my body, and it will start to translate into an amino acid sequence that looks exactly, if not identical to, at the end of the spike protein in the virus. And then it's going to kick off my immune response, which is going to start making a lot of antibodies. specifically against the end shape of the COVID-19 spike protein, but I don't need to get the virus to do that. All I need to have is the RNA sequence to inject into me, and my body will make the thing automatically, and I'll get the immune benefit from it. Cool.
Do each of those degrees like correspond to each of the levels? Like the 1, 2, and 3?
Primary protein sequence is the sequence of amino acids. Remember that? From days, how long is like 1 million years ago? That was Thursday. Sequence of amino acids, which what we have, that's what we just came up with. We just determined primary protein sequence from the RNA strand that we got from the DNA strand. Secondary are the coils and folds. Tertiary structure is the three-dimensional shape arrangement. There's a quaternary when you get different three-dimensional things that interact. And I think we were talking about hemoglobin as an example of that. That's not familiar? Vaguely. Vaguely. So how would you describe what DNA does? It just, I mean, you don't build proteins off of it. What you do with the uDNA is you use it as a template for RNA. RNA, you then use as the template for amino acids sequence, which you then have codified in the codon table, which represents one of the pieces of evidence that we use that indicates that all life on Earth evolved from the same common ancestor, because every single living thing on Earth uses the exact same codon table with no variation. It's like, what are the odds of the exact same relationship happening twice independently? It's like 0. So it's like, well, everything on Earth has that exact same relationship between RNA and the amino acid that it codes for in the transfer. So it's like, well, that's unlikely to happen the same way twice. So maybe all life did come from a common ancestor, which is kind of neat.
Like everything, including plants and stuff like that.
Word. Yeah. I mean, we're talking about the way back. So this, going from DNA to RNA, is called transcription. Going from RNA to the amino acid sequence is called translation. This process here, going from RNA to amino acid, happens on the surface of something in your cell called a ribosome. It's one of your organelles.
Wait, say that again?
Ribosome?
No.
A ribosome?
No. Like, why is
It happens on the surface of the organelle on which this happens. Translation happens is on a ribosome. So a ribosome is like the workbench on which you take messenger RNA strands and assemble the proteins. So which one is the blueprint? Well, it's really not this one. It's more like the RNA is actually the blueprint. This is like the hard drive. It's like long-term energy storage.
Did you explain what a ribosome was?
Yeah.
I just kind of like completely skipped out on.
I get it, man. It's cool. You're tired. You stayed up all night texting. She did. We talked about this earlier. It's the surface, right? It's an organelle. It's very small in your cells. And it's the surface upon which the protein is constructed. So it's literally like a workbench.
Okay.
Do you remember back when you were taking bio in high school? Anybody who took bio in high school? Anyone? You've heard of rough ER? Yeah, rough and smooth ER, these organelles. Remember that vaguely? If you do, no need to. We'll get into this in a week or two, like more of these organelles. Rough ER is rough because it has a lot of ribosomes on the outside of it. That's what makes the rough. That's what makes it rough looking. So it looks like it's got a roughened edge under a microscope. It's all the ribosomes that are on the surface of that organelle. So if you have a cell in front of you and it's mostly jammed packed with rough ER, it's jam packed with a lot of ribosomes. What is that cell doing most of the time? It's making proteins. It's making proteins. So it's like, think muscle, think the lining of your stomach where you make a lot of pepsin, and you wear in your body where you make a lot of protein and repair a lot of protein. It's going to have a lot of rough ER in it because it needs to have a lot of ribosomes for the translation to happen. So we can start to get to the point where, and smooth ER doesn't do that, right? So we're starting to get to the point where we can make these associations between what are the function of these biomolecules, upon which surfaces do these reactions happen, how do we determine what a cell can do based on the concentrations of these different larger structures we see inside of them, and where can we make inferences about, or if I needed one. So if I had a needle like that long, and I'd say, hey, can you come up and lay down on the table for a second? And I say, okay, so where do I harvest cells from to get a lot of rough ER? You would say, where should I poke? If I wanted, if I was like, hey, we're in lab, we're going to make a slide and it's going to have a bunch of rough ER on it. Where do I harvest those cells from? Yeah, muscle tissue, stomach lining. Where else do you make a lot of proteins? All over, some places, but not so much in others, right? If I wanted, anyway, we can get into this more about cells, but you can do this like throughout the body. It's like kidney cells are going to have a lot of epithelial tissue, your liver is going to have a lot of smooth ER, making a lot of detox kind of stuff. I mean, so we can start making sense about it. So which molecule is like where the action is? So DNA is like long-term energy or long-term information storage. Not too exciting. This is the end product. The real cool stuff happens with RNA. That's where the action is. What is the actual basis of the codon table? So when we see AAG, how do we relate that to lysine? This RNA strand that contains this information is called messenger RNA. What relates the units of messenger RNA and corresponds them to the amino acid is referred to as transfer RNA. The transfer RNA molecule, it's a protein, and the top half of it is going to be complementary base paired to the messenger RNA. So the top half of the transfer RNA is going to be AUC. The bottom half is going to be a protein, kind of marker on it, that specifically attaches to lysine. So we talk about, hey, there's DNA and there's RNA. It's like, well, with RNA, there's like messenger RNA, and then there's pre-messenger RNA, and then there's transfer RNA. RNA does a lot of neat stuff, which is why we think of this entire system here that operates. It's like, that's actually kind of complicated. When we think about how does life emerge on the early Earth, what pieces do we have to have in place and how they operate? It must be complicated. The thing that is central to this entire operation of your cells and life on Earth functioning is RNA. So a lot of people think because of that, some of the very first molecules that existed on the Earth that were able to do interesting things were RNA nucleotides, RNA chains, which is kind of neat. So it's like if you want to search for how did life emerge on the early Earth, not a bad place to start. How does RNA work? Because it's central to the whole operation.
So the RNA itself is mRNA?
Is what?
Is mRNA?
When a strand of RNA is coded off the DNA, we refer to it as messenger RNA. We can't just call it RNA because there's also transfer RNA. So this is like the mRNA, the messenger RNA confers the message from the DNA. The transfer RNA relates that RNA to the amino acid. The basis of this transfer RNA molecule is That's what gives you this. So for each one of these, there is a transfer RNA molecule that makes that relationship. What else do you see that's interesting when you look at this codon table? You have 4 first base, 4 second base, and 4 third base possibilities. You get the possibility for 4, 16, for 64 different possibilities. Correct? 4 to the third. 64, yeah. But you don't have 64 amino acids, you only use 20, right? So there's some redundancy here. Anything that starts with a CG is going to be arginine, right? It doesn't matter what the third base is. They're always going to be the same. Anything that starts with a GG is going to be glycine, right? CC is going to be proline, right? And what you, I mean, this is all over the place in here. There's very few that are just like one and dones, except I think tryptophan is the only one that is like, exclusively that thing is tryptophan and nothing else. What this tells you, the way this looks, is that the consequence, and this is where people are talking about how bad is the mutation if you have one, like if you have a miscode, they say, well, it's not great, but it also depends. A lot of times, if you have a mutation in the third codon, a lot of the times that is actually not going to matter, right? It's just, you're going to get the same thing anyway. You know, freakish appendages don't come out of our bodies in strange places because we have a miscode in our third codon. It's like they don't have consequences for the protein usually that's produced. Sometimes they do, but a lot of times they don't. However, a miscode or an alternative code in the very first, and a lot of times the first and the second one, will have consequences. So if you think about it, it's like, well, a third unit codon mutations usually not going to make a difference, right? So it's like a third of the mutations that you have are sort of meaningless anyway, right? Because it's like, it's, oh my God, I have a mutation. It's weird. It's like, yeah, but it's in the third place of the codon, so it's just going to be, it's sort of neutral. It doesn't have consequences for the amino acid, which ultimately that's where the consequences are. Make sense? Makes sense. It's like the first one always will, if there's a change. The second one usually will. The third one rarely, but occasionally does. Questions, thoughts, comments? What do you think? Makes sense? Questions? It's on my corn joke again.
It's too corny.
Too corny jokes. It's the corniest joke I have. Even that was so stupid, it was fun. Will you have to, throughout the semester, transcribe and translate DNA into amino acids? Of course you will. I mean, it's the perfect set of quiz questions that you could ever possibly. I wouldn't be surprised if you had a DNA strand and a codon table on Thursday placed in front of you. I would find that a reasonable expectation. And we will probably do that at 9:35. How'd it go this morning?
I came early, okay? I didn't drive.
You're driving in, you're gonna say, damn it, I'll show him. I'll be there at 9:34. Son of a bitch. See, I'm here. So we have DNA. Great, perfect, we're there, awesome. I'm talking about a eukaryote. It's in the nucleus. It's in the nucleus. Talking about a prokaryote, which is the bacteria on the archaeans. It's referred to as a nucleoid. So the bacteria and the archaea domains don't have a nucleus, but they have a structure called the nucleoid. So what the nucleoid is in bacteria and archaeans, it's like you take the DNA and you like wad it up into a ball and you shove it in the corner. That's it, right? So it's not separated from the rest of the cell by any kind of membrane or anything like that. In the eukaryotes, which is fungi, plants, protists, things like that. It is
like the
partition.
difference between single-celled organisms and like multi-celled
It's not. It's not. There are single-celled eukaryotes, there are prokaryotes that act like multi-celled things sometimes. Blue-green algae, for example. So I wish it was as simple as that, but it's like, sadly, it's not. They're extremely complex prokaryote things, are extremely simplistic eukaryote things. I want things to be easy like that, but they're just not in bio.
What's the word next to bacteria?
archaea.
Archaea. We have RNA, correct? That's double-stranded. This is single-stranded, and there is messenger RNA. This transfer RNA. Bless you. Who's going to go on from here and take cell or genetics? Any bio majors or pre-med or anything like that? Anyone? But never mind. I was going to say, if you do, there are others. There's a couple others. There's pre-messenger RNA. There's some details that you'd go into. There's a couple other different kind of RNA sort of kinds that are just like that cell biology stuff. We've exhausted our functional use for what we needed to do. And talk about this though. I mean, it's important for us because saying, hey, is anybody into bio major, or is anybody going to pre-med or anything like that? And like, nobody raises their hand. Really, really, really? I get that. That's fine. But I mean, why is this relevant? I just got injected with this last Friday, about a week and a half ago, right? Pfizer loaded me up with an updated messenger RNA strand specific to the Omicron spike. And it got injected into me. So you know what? Maybe it ain't a bad idea to have a little bit of a passing knowledge of why messenger RNA is important. So go ahead.
Are the two things under DNA a part of it, or they're just separate? I just wanna know.
This
Yeah.
DNA in eukaryote, there are two kinds of organisms on Earth, eukaryotic, that have a nucleus, and if they have a nucleus, the DNA is in the nucleus. The prokaryotes, the bacteria, the archaea, they don't have a nucleus. So it's, but you can see it under a microscope, the DNA.
Gotcha.
It's one in the corner and it's referred to as a nucleoid. So if you ever say you can see the nucleus instead of a prokaryote, wrong. But the question would be, back in the day, how could Pfizer and Moderna knock out a COVID vaccine in under a year? Right? I mean, that was unheard of. But what we find out is that with this technology and knowing what messenger RNA does and how it works, you don't actually need to grow the virus. Like back in the old days, it's like you'd have to either get like eggs or some other biological media, grow the virus, harvest the virus, destroy the virus, inject the destroyed virus chunks into your arm after some refinement. Really intensive process, really took a long time. Now, right, thanks Moderna and Pfizer BioNTech, all we have to do is know the RNA sequence of the spike. Know the RNA sequence of the spike, you can put it in a vat in Kalamazoo, Michigan. You can code the messenger RNA as many times as you want, put them into little lipid spheres, put them in a needle, and you're good to go. I mean, Omicron only popped up. I remember when it did. I was actually in Kalamazoo family in Kalamazoo, which is where the Pfizer production facility is, right there. And that was Thanksgiving-ish, 21, Thanksgiving 21. Here it is, September. That was the very first time that people started to use the word Omicron. It was like, hey, Delta's gone. There's this other weird variant that might be popping up over here a little bit somewhere, but I'm sure it's not bad and it won't do too much to us. Right? And then under 7 months later, you already have vaccination production on scale. So it's like most of your vaccines that you get from now on, including a flu shot, will be these mRNA viruses. Because it's like expense goes down by 90% because you don't have to have these big, I need to actually grow the virus things. It's like you can build it out of nothing. All you need to do is have a data file that has the sequence in it. It's like, great, that's all you need. Perfect. Just need one person to sequence it and upload it, and you're good. And you can find those spike protein sequences on GenBank. Download those for free if you want. You can do this in your garage. So by Thursday, come to class with fully transcribed, translated, coded vaccine for influenza in a syringe. Go ahead.
So, with there being like different types of vaccines for COVID, like Pfizer, Moderna, et cetera, and some having malfunctions on people in the beginning of them, is that the
Malfunction.
like just different effects that I guess cause different stuff, like with certain shots, example like Johnson & Johnson at one point. Is it the mRNA that affected the strand that caused this? Or in the vaccine?
It probably not directly. It's probably one of two things. Either everybody in this room's immune system is different,
Right.
and sometimes they're very different. Sometimes a vaccine, and this is not unique to the mRNA ones, can trigger part of the immune response where it doesn't react well. And sometimes it's no more than just an allergy to the solution that's the basis of the vaccine itself. And this goes back to like old flu shots back in the 60s and in the 50s. I mean, there are some people out there that are allergic to eggs, like albumin. Like you have an immune response to eggs. Sadly, right, a lot of the eggs, chicken eggs, do a very good job of hosting and producing viruses as well. So they're using eggs for like viral media to replicate the virus in. And you get the shot, and it's like, oh my God, immune system is freaking out. You just got like egg albumin proteins injected in, right, from the media that the virus was grown in. So usually it's not the thing itself. My first COVID shot was fine. My second one, like of the primary series back, I had 104.5 degree fever for about 3 hours. I was tripping out. It was one of those like shamanic things where it's like, boy, these are some really creative thoughts I'm having right now. That's awesome. And it's like all I could think about. And it's like I worked it out. You want a freaky story?
Yeah.
Here's a freaky story. So I get this shot and it's like, whew, I'm off to, it's like exactly 12 hours to the second. It's like, I'm going to go to bed. It's like, all right, I'm going to bed. I walk up, and when I got up to go upstairs, I was fine. Not 45 seconds later, when I got to the bedroom, it's like my fever was at least 102, the full on body aches, the whole shebang. And I lay down and, once your fever starts hitting 102, you stop feeling hot, you start feeling shivery. So it's like every blanket in the house, pile on me, please, because you stop thinking clearly, because that is not the thing to do when you get a high fever. But what am I going to do? And so it's like, take my blanket and I'm going to cut somebody, so it's like I'm just covering myself. And we have one of the digital laser thermometers. So, I'm like, honey, I'm pretty hot. Can you take my temperature? And she's like, beep. And it's like the little thermometer, it's like it's flashing red, it's beeping, and it's like, oh my God, it's like 104.7. It's like, that's hot. Wow. And so she goes back to sleep because whatever. And it's like all I could do, because it's hard to sleep when you have 104.5 degree body temperature. And did she offer to get me some ice water? No. Did she offer to give me some acetaminophen? No. It's like, where were you with all this? Why didn't I get up and take something? I would cut somebody for a blanket. Why don't you get up and give me? Anyway. And so all I could think about, because my brain was getting a little cooked, was how to effectively and appropriately price sounds that we hear in our environment for an exchange market. So if we could build an economy off of sounds, what is the value of a sound that we hear? And how do you price it? And what would cause fluctuations in the market for that? And it's like I had the complete and total effective, fair market monetization for sound, like in my head. And it's like, it all makes sense. This is amazing. And it's like fever went down and it was all gone, you know? And a couple of days later, I was talking to this lady down the street from me, and she's super smart. She works for NASA, and she got her second Pfizer shot like right at the same time. So I said, "Hey, got the shot? Yeah, I got the shot. Yeah, it was pretty rough side effects, wasn't it?" "Yeah." She said, "Yeah, I went to bed and all I could think about was economics." Freaked me out. So it's like, is there something about the Pfizer primary series that like reacts with part of our body somewhere that fires up I'm interested in economics now and free market monetization of obscure things? Because like she was up late at night, it's like all you could think about is economics during that high fever. Isn't it the wildest thing? Ain't that the wildest thing? And no, the messenger RNA is not going to implant itself into your nucleus and become part of your genome, and you're not going to turn into a hippopotamus.
So every shot you get has a different RNA in it? I mean,
The first batch did. This is the first batch of the COVID shot that has a different messenger RNA strand in it. And it's only a little different, but it's specific to the Omicron variant. Right, the original series is like Alpha, but they didn't update it to Delta because it works equally well with the Delta variant, which was because there was a big question about whether or not we have to update it for Delta. It's like the Alpha one works just as well. It's not that big of a difference. It's just a little more lethal. Just a little more lethal. The Omicron one, though, it's not as lethal, but it's super catchy. And the spike is actually different enough that the original series doesn't work very well against it. So it's like when you get the Pfizer or the Moderna updated one now, it's about a 50-50 of like half the messenger RNA is for the original Alpha, which works in Delta, and some of those old strains are still out there. And the other half of it's Omicron specific. And a lot of people think that Omicron is the end because like it might be as good as the virus could actually get. And it's just going to become endemic and poop out. There might not be any more variants of significance after this. It's like it's like back in the day throughout the 20th century, the world record time for the mile, like for running for the mile, went up very, very quickly and then it kind of plateaued out. It's like there's not, and now it's like the difference between the top 4 fastest people in the world is like at the 100th of a second. You know, it's like it's really, really hard for a human to get faster than Usain Bolt. It's just not. It's like we're at these structural limitations of how fast a human can go at that point. The COVID virus might be close to the structural limitations of its transmissibility with what Omicron is doing. It might be natural selected out in terms of like it's as good as it can get. Might not be anything else that it does that's meaningful, because the variant doesn't want to be deadlier. It wants to be more transmittable. You know, the deadliness sort of goes the way that it sort of does. But what we see in COVID is very similar to the way that a lot of viruses go. They get more transmissible and they get less lethal. You know, that's always the way that it goes almost. Because like if it gets like more catchy and more lethal, it doesn't spread as much because when you get it, you die, right? And it's like it kills its host. It's like, so they tend to get more transmittable and less lethal. So now we get like an annoying little sniffle, little scratchy throat for a couple of days. 50 years ago when that virus showed up, it killed half the population of Estonia, you know, or something like that. They get more catchy and get less lethal, and COVID is doing exactly that. It's doing exactly that. Go ahead.
What are the structural differences between the nucleus and the nucleus?
What?
Like the nucleus and the nucleoid, like are there I'm just curious, are there any major structural differences?
A couple. Let me go back. Yeah, there are. Eukaryotes, the DNA. It's linear, so it's straight, long line, beginning and end. 46 strands of it, 23 from mom, 23 from dad. More about this later. Your parents had sex. Oh, it's coming. Give it about a month and a half. Right? We can get there. And the DNA is wrapped. It's kind of double coiled, and these little proteins called histones. So it's like, think about a long string of yarn. It's like double wrapped, double wrapped, double wrapped. So it's like it's got a protein kind of anchor to it. It's called a histone. Prokaryotes, bacteria, archaea. DNA is circular, so it's like a hula hoop. But in archaea, their DNA is also wrapped in histones. So it's thought when you have a large tree of all things on Earth, life starts here, codon table starts right here, right? We have the eukaryotes, archaeans, and the bacteria, they're the weirdos far out. Histones evolve here. Nucleus evolves here. Time goes that way. So we think the archaeans are more closely related to us than the bacteria are because we both wrap our DNA around these histone proteins. Just kind of neat. Within the eukaryotes, you have these protists. We have the animals, animal kingdom, our first cousin, of the fungi, which is kind of neat. Plant lineages either come out of the base of this tree, but more likely, actually, independent base out of the protist tree. Right, but first cousins, fungi, a lot of stuff in common, which is fun. Questions, thoughts, what do you think? How was Nucleotide Day? Good? So what's going to happen here, the point of this whole conversation was this. Because now we have this process of how do we go from nucleotides that you have in your, or DNA that you have in your nucleus to a three-dimensional functioning protein. These proteins are going to catalyze reactions. And when they do that, we're going to call them enzymes. And so on Thursday, we'll talk about how enzymes work. How do they not work? How are they regulated? How are they controlled? What kind of reactions are we talking about? The rules for how enzymes function. And then in lab, we'll look at some enzymes and see what they do. Perfect? You got a day full today, Brooke.
Yeah.
How you feeling?
I'm gonna go home and not think about it, so it'll process later.
That's best. That's best. If there's a skill set for Thursday that I would recommend, it's like being able to take a DNA strand and make a protein from it. 1, 2, 3. Bye, everybody. Green. Bye. Oh, yeah, I'll take homework if you got it. Honestly, I don't think about it that much. It's more for you than it is for me. Yeah, fancy, isn't it? I love that. I think it's fantastic. Thank you, David. Thanks. Oh, Sylvia, you got a board full today, didn't you? What do you think about all that? I'm sure you learned all this back from that. Yeah. It all comes back.
So I just put like a.
Yeah, I've done that before. Yeah, you're good. And that myself. Thanks, take care.
Hey, I had a question.
Sure.
So when we were looking at the transfer between DNA and RNA
Uh-huh.
and just sort of corresponding the letters to each other. So I just want to make sure I understand this. I think I've got it, but I want to make sure. So obviously with each of the letters you have the opposite where you pair it like that.
Right. With DNA pairing.
DNA pairing. So with
Right.
RNA pairing, the T in this one would always be a U, correct?
The A will always be a U. The T will always, the DNA T will be a,
The T will be an A, but whenever it's
the
an
A
A,
will
it
be a U.
transfers.
Correct. That's correct. That's correct.
Okay. I just want to make sure I get that written down.
Good to get it right now and get that